Good morning, professor and classmates. My name is Yeonwoo and I'm studying in the genome editing lab. Today I present about the plant genome editing and our lab research process. As we know, now we are facing so many problems. World population growth is increasing very fastly, and environmental problem also affect to our food production and available land also reducing. So we need to develop the new plant breeding technology. It should be more efficient. Conventional breeding have some limitation. First is limit to exchange between the same or closely related species. Second is little or no guarantee of any particular gene combination from millions of crosses generated. Third is gene linkage. If some gene placed physically near the other gene, so difficult to get the plant which include our desired target gene only. And it take a long time to achieve the desired result. And other mutation breeding, transgenesis, cisgenesis also have some problem. Genetic modification by random mutagenesis also cannot be controlled. We cannot control the gene insertion site or side effect like that. And random mutagenesis require manpower to select the desired phenotype. So we need to develop new plant breeding technology. So nowadays we are using CRISPR-Cas system. So before explain the CRISPR-Cas system, we need to know sequence-specific nuclease. So there is 3 generations of sequence-specific nuclease. Is in the genome editing, we are using this one as a genetic seizure. So first generation is zinc finger nuclease, is first artificial restriction enzyme generated by fusing zinc finger DNA binding domain and DNA cleavage domain, is POC1 nuclease. And second generation is TALEN. TALEN also fusion protein, is TAR module and POC1 nuclease fused. And third generation is CRISPR-Cas. So before that too, zinc finger and TALEN require protein engineering, because to recognize specific DNA sequence, they require to modify that recognition domain, is zinc finger and TAR module. So every time, if your target gene or target location changes, so you need to resynthesize that protein. So it requires money and a long time to synthesize. So, but CRISPR-Cas system consists of Cas protein and guide RNA. So in this system, Cas protein and guide RNA, usually guide RNA guide the protein to target sequence. So just we synthesize the guide RNA only. So this one is cheaper and easier compared to protein engineering. That's why nowadays we are using CRISPR-Cas system better than other two. So yeah, CRISPR-Cas system come from the prokaryotic defense mechanism. Actually, when the phagy or other virus impact the bacteria, when they insert their DNA fragment into the bacteria cell, and bacteria usually express the CRISPR array and Cas protein, and they generate the matured guide RNA and Cas protein, and that cleave the invaded DNA or RNA. For the genome editing, we engineered the native CRISPR system. So we linked there's two guide RNA, CRISPR RNA and transacting CRISPR RNA, and usually we optimize the codon for our material. This two is representative Cas protein. First is Cas9 and second is Cas12a. Is CPF1. The two have many difference. Cas9 usually require G-rich PEP, NGG, and it include RUBC-like domain and HNH domain. And because they have two different nuclease domains, so Cas9 can be used as a leakage. We can make a mutant version which delete one nuclease domain. And it requires CRISPR RNA and transacting CRISPR RNA, but we usually put the linker and just express single guide RNA. And Cas9 usually generate the blunt end, and it cut the proximal of the PEM. Unlike Cas9, CPF1 require T-rich PEM, and it have two RUBC-like domains, so difficult to make mutant version like leakage. And it requires only CRISPR RNA as a guide RNA, and it generates sticky end, and it cut the distal of the PEM. So for the purpose, you can choose preferred one. After generate double strand break, DNA double strand break, the DNA follow the DNA repair pathway. So there are several pathways, but usually we distinguish NHEJ, non-homologous end joining, and HR, homologous recombination. So for the gene knockout, we usually go to the non-homologous end joining. So actually we cannot know they follow which one, but usually non-homologous end joining generate indel mutation. So in prime, if indel mutation happen in prime, so that GDS region cannot generate perfect mRNA and cannot generate protein. So we can get the loss of function mutation. And for the gene insertion, our plant should go to HR pathway, but problem is first it requires template. In the native form, usually sister chromosome is homologous DNA template, and HR pathway is very low efficiency because almost in the cell cycle, almost DNA double strand break is very serious problem. So plant cell and animal cell want to repair fastly, so NHEJ is more faster than HR, but it have so many error. So in the some stage, cell usually go to the HR pathway. So many scientists want to improve this HR efficiency. Later I explain more. So this one also depend on the according to your purpose. So if your plant material is monocot, you can use monocot promoter, or dicot, you can use 35S like that. And you can choose the Cas protein. For example, for the HR, usually we are using CPF1, and for the indel, usually we are using Cas9. And after that, we need to choose the plant selection marker, hygromycin, kanamycin like that. and design or construct. After construction, usually we are using Golden Gate cloning system, make a vector and check the sequence by Sanger sequencing. After confirm the construct sequence, we need to plan the plant tissue culture. So need to decide media type, culture condition, selection marker concentration, hormone concentration, like that, and go to the plant tissue culture. So maybe several months later we can get the plant and need to check editing region. So to check the genome editing result, we usually send Sanger sequencing. Nowadays we are using NGS also. So first extract the genomic DNA from that mutant and amplify that target gene region, and we design the primer near the guide RNA and check in there. But in the Sanger sequencing, it's difficult to observe some low efficiency of in there mutation, so maybe NGS is better. And in other case, we can treat some chemical. For example, if you generate salt tolerance plant, you can add salt into your plant germination media and you can compare with wild type. Or if you generate drought tolerance plant, you can test that one also. And other thing is just extract the DNA and digest using some sequence specific restriction enzyme. So if in their mutation delete that restriction enzyme recognized sequence, so you cannot get double band. So in this gel figure, you can see the wild type only. There's 2 band, but in the HR allele, you can see only single band. So the 3 band may be some contamination or hetero, but we can assure by this one, so we need to send sequencing anyway. Yeah, until here I explained the some generate commercial crop, because it's all about the editing method. So if you want to generate some commercial crop or some commercial or you're interested in mutant, you can use that way. And in our lab, we try to develop some novel editing tool. So already I referred this one. HR efficiency is very low compared to non-homologous endojoining, and HEJ is only 1%. And many people want to improve this efficiency, our lab also. So I will introduce one short brief information. So already we know the important factor for HR mediated genome editing for gene logging. So first is the number of donor templates, because in the native DNA repair pathway, sister chromosome play the role as template, but in the genome editing we can provide the template. So if we increase the copy number of template, that give the high chance to meet double strand break DNA and template. So it's very important. And second is, anyway, HER pathway require double strand break, so high amount of double strand break is good for HDR. And third is cell activity, because HDR requires so many associated enzymes. So usually this pathway happened in G2 and S phase, and so this cell cycle is also important. So we are using Keras for HDR. Actually, yeah, for the old transformation, but we sometimes we treat some chemical for this cell cycle like that. And already reported this one, Gemini virus can reprogram the plant cell from non-dividing cell to dividing cell. That means inactivated, actually not 100% inactivated anyway, inactivated cell to activate cell. So we are using this information, so we use this replicon, viral replicon, for the genome editing, but it includes some movement protein, code protein like that, so we remove that tool and we add editing tool. So this is that model replicon system for genome editing. So we remove the code protein, movement protein, and add Cas9 and guide RNA and other plant selection marker, and transform the plant cell. So we can transform Agrobacterium or Bombardment or dipping like that, so many ways. It's just you can choose some preferred method. In our lab, we are using Agrobacterium mediated transformation. And when the introduce this vector into plant cell, they can make the circularized form. So that LIR is origin or replication, so they can replicate and increase the copy number. So this means we can provide high copy number of template, and we can express very strongly the Cas9 and guide RNA. So it generates good amount of double strand break and can provide the high copy number of template. So we are using this way. And to assess this system, we usually doing the PCR. So in the linear form, primer direction is opposite, so cannot amplify any region. But when they make the circularized form, we can amplify that LIR and SIR region. So you can see the band, target band around 500 base pair, and it should 3 to 12 days after transformation. So we can sure our replicon expression system working very well. And later we need to check editing efficiency. We need to apply this system for editing. In summary, our lab focus on the development of commercial crop and novel editing tool, and we expect this technique and crop will help to our food production and food security. It is all of my presentation, and thank you for your attention. Thank you.